Okay, so we are right at the top of the hour. It's 2 p.m. Here, and I just want to take a moment to welcome everyone to the webinar and thank you all very much for joining us today. We're excited to have you here. My name is Kayla Ripple, and I'm a senior associate with the Lenfest Ocean Program. So for those of you who don't know us, the Lenfest Ocean Program is a grant-making program that funds ocean and coastal research projects and expert working groups to address needs that are facing decision makers and stakeholders today. You can learn more about us and the projects we fund at lenfestocean.org. And while you're there, you can also sign up for our newsletter. And if you're on Twitter, be sure to follow us at lenfestocean. In fact, we'll also be live tweeting this webinar on the hashtag LOPwebinar. So feel free to use it too if you'd like to engage with us there. We're excited to have joining us today Dr. James Morley with East Carolina University, Dr. Verena Wang, also with East Carolina University, Dr. Lorenzo Ciannelli with Oregon State University, and Mr. Mark Nelson with Lemmy Nation and previously with Noaa Fisheries. These are just a few members of the research team that's working on a project we're supporting to investigate the role of habitat and life history in fisheries vulnerable to climate change. This project started in September 2020 and is an outcome of an Ideas Lab that we hosted in 2019. As part of the Ideas Lab, Lenfest and our partners, the David and Lucille Packard Foundation and the Biodiversity Funders Group, convened researchers, managers, and stakeholders to generate research priorities around fisheries management challenges associated with shifting marine species. The workshop was focused on the U.S. East and West Coast and has since resulted in a series of projects that will advance understanding and contribute solutions to different aspects of the climate and fisheries puzzle. Scientific efforts to project changing fish distributions have advanced rapidly in recent years. This project will build on those efforts by including life history traits and habitat requirements in models to determine where fish may go, at what stage, and if their habitat needs may constrain their movement. I'll leave it to the researchers to get into the specifics, but for us at Lenfest, we see this work as an important building block that can help fill a knowledge gap in understanding how species move and shift, which in turn could help managers and stakeholders make more informed decisions as they work towards climate adaptive fisheries management and strategies. This project is still in its early stages. In fact, this webinar is what we like to call a launch webinar, which is meant to inform and engage audiences from the outset of a project, rather than just at the end when a project has results. So if you're interested to engage or have questions, I definitely encourage you to reach out to either us at Lenfest or to members on the research team. Before we get started, I just wanted to share a few webinar logistics with everyone. With so many people in attendance, we have all attendees muted to prevent any feedback or echoes. There's going to be time at the end for questions, so please use the Q&A panel to type and submit your questions at any point during the webinar today. We'll keep track of those in the queue, and then at the end, I'll read aloud the questions for the researchers to answer. Now, depending on how many questions we have, we may not be able to get to them all, but folks are certainly welcome to follow up with us here at Lenfest or with the research team. And finally, we are committed to making Lenfest funded work transparent and easily accessible for all stakeholders. So we'll be recording this webinar and we'll distribute the link broadly afterwards. Feel free to share that link with others who are interested but may not have been able to attend today. And as this project goes on, we'll continue to share information on its progress via a variety of avenues like on our website, in our newsletter, on Twitter, and other places as they pop up and seem appropriate. But for now, I've started creating a running distribution list for everyone who's interested. If you're not on that or unsure if you are, just send me a note. You can find my email on the slide right here in front of you. And lastly, after you close out of the webinar, you'll be taken to a very short 3-question survey. Please do take a few minutes just to fill out that survey. We'd be so grateful for any feedback you can provide. It'll be extremely helpful for us to better understand the best way to engage with you and with others as we continue outreach around this project. And with that, we'll go ahead and get started. And I'll turn things over to Dr. Morley. Jim, I'm going to pass the presenter privileges to you.
Okay. Yeah, thanks, Kayla, for that introduction. And thanks, everybody, for tuning in today. We appreciate the time you're taking. So Kayla summarized our study pretty well. Our work is going to be focusing more deeply on mechanisms behind species responses to climate change and climate variation, and more specifically, looking at how life history, species life history, and life stage specific habitat requirements might affect a species' ability to respond to climate change. And so, yeah, as Kayla mentioned, we do have a big team, and we're a geographically broad, diverse team. That includes myself at East Carolina University, along with 2 postdocs. Verena Wang, who's going to be speaking after me, is the primary postdoc for the project, and Leela Schlenker is going to be putting some time into it as well this year. Matt Ajamian at Harbor Branch, Florida Atlantic University, Stephanie Archer at LumCom in Louisiana, Marissa Baskett at UC Davis, Lorenzo Ciannelli at Oregon State, Emmett Duffy at the Smithsonian, and Mark Nelson at working with the Lummi Nation. And previous to that, Mark worked with Noaa Fisheries for many years. So just a kind of a broad overview of species shifting in response to climate. Broadly speaking, climate change is acting by shifting the geographic locations of the preferred temperatures for each species, and we often refer to that as a species thermal envelope. And generally, species have been shifting their distribution to keep pace with their thermal envelopes as they change. And this is a figure from Malin Pinsky and colleagues that came out in Science years ago, and this sums it up very well. So in this figure, each of these dots is representing an individual species, the rate at which it is shifting latitudinally over time, and the rate at which its preferred temperatures are shifting over time. And you can see there's a strong correspondence between these two things. But there is variation about this relationship. There are many species that are shifting more rapidly than we might expect, and there are many species that are shifting much more slowly than we might expect. And this has been a really exciting field of marine ecology. Really, it's developing very fast, and there's lots of super smart people that are working on these questions. Some of the key things that we've learned in the last 5 or 6 years are that the rate and direction of species shifts can vary depending on your spatial scope. So if you zoom into a species, a subset of its range, it might shift in a more unexpected direction. So it's not a uniform response change. Also, species relationships with climate can be complex. So they might respond to a lagged temperature effect. For example, winter temperatures might be more important than temperatures that are measured at the time of sampling, for example. And overall, it hasn't been easy to link species life history traits to their response in climate change so far. But we think that if we delve a little deeper and look at species habitat requirements and some of the life history and life stage dynamics, that we can figure some of these questions out. So as a species thermal envelopes, as its preferred temperatures shift across a landscape, the more static features of a species habitat will change considerably in some cases. So this is one example for the Northeast continental shelf, and what you're looking at here is the seafloor habitat overlaid bathymetry and sediment characteristics. And hopefully when you look at this figure, you can see several very distinct habitat zones or regions, for example, the continental shelf break or Georges Bank. And individual species may have very strong associations for some of these regional habitat characteristics. Again, Georges Bank would be a great example here. And those strong associations we would expect to have a major impact on how that species will respond to a shifting climate. And while I'm showing, very static features here in terms of benthic characteristics, this also would apply to more dynamic habitat features, oceanographic features like the Gulf Stream position, or on the West Coast, factors associated with upwelling. So in addition to that notion of these static habitat features, it's also been illustrated. Well, this is something we've known for a long time, but there's been some great papers out recently that have maybe reminded us of this again. And that's that distributions of species vary across life stages. And often this is viewed as shifts in ontogenetic shifts in depth. And here's an example with summer flounder. On the left side there, you're looking at locations of summer flounder trawl catches from the groundfish survey. And this is in the spring at 2 different size groups of fish. The smaller fish you can see in the red tend to be closer to shore in most years. This is aggregated over a number of years, and also shifted further or more to the south of the overall range. Whereas larger fish tend to be associated with a shelf break and are generally farther to the north. In a recent paper by Peretti and Thorson, they tracked the shifts in the distributions of these 2 size groups. And that's what's shown there on the right, and the colors are corresponding to the size groups. You can see the differences in the spatial preference of these 2 size groups. And overall, the smaller fish were shifting to the north about twice as rapidly as the larger fish. One more great example that came out very recently from a group of folks at the Northwest Fisheries Science Center. They used an acoustics data set on Pacific hake, and they examined how this species responds to temperature variation across space and at different life stages. So on the left side is the smaller group of fish, age 2 fish, and what the take home here is that the smaller fish tend to have a higher biomass when the temperatures are lower, and it's a little bit more uniform across space. That's what that blue color is indicating. Whereas the larger fish have a more spatially varying response to temperature, where the areas in the red are showing where this species has a higher biomass during relatively warm years, and then the opposite effect during relatively cold years in the blue. So the point I'm trying to make with these examples, all 3 of these examples, is that we think it's difficult to fully understand climate responses of a species without more detailed understanding of these spatially explicit responses of a species to climate across different life stages. So there's a number of challenges from distribution shifts, and Mark will talk a little bit more about these later in the webinar, and I'm sure many of the folks have tuned into this webinar because they're aware of these challenges, but maybe a couple you haven't thought of. One is interpreting data on species shifts can be equivocal or uncertain. For example, if a species is shifting very slowly or much more slowly than expected, is this indicating some sort of spatial habitat constraint at specific life stages? And should that be a concern for that species' ability to adapt to climate? And second, adaptive management strategies can be challenged. And I think this is an important point that, in my thinking here, is more from a stakeholder perspective, that climate adaptive policy might be harder to implement if we don't really have a detailed understanding of the mechanisms behind a species shift. On the right there is the schematic from the Noaa climate science strategy, and I've included that just to show where our work is feeding into with Noaa's objectives. Several of these tiers here, including providing early warnings and tracking changes of different species, certainly understanding mechanisms for species responses to climate change, and then how to better interpret and how to better conduct forecasting into the future for different species. So our work is a 3-year project, and we are progressing through 4 phases, we're calling them. And the first phase is a more qualitative assessment of habitat constraints based on a previous example. Here we're looking at habitat limitations or changes due to climate change across species life stage, and also potential issues with life history connectivity between life stages for a species and how climate will affect that. Phase 2, which Lorenzo will be talking about in a bit, is looking at spatial modeling across different life stages and looking at these principles of how spatial differences of species across life stage respond to climate. And then finally, looking at climate change exposure at different life stages and future projections across life stages. And then finally, finishing all that up with a larval tracking model where we're looking at how comparing present and potential future distributions with connectivity of adult spawning with key nursery habitats. And overall, as we progress through these 4 phases, we are following this general graphic on the right side, where we're starting with many species and with a qualitative analysis, and then when we finish up with the larval tracking, we're probably just doing that to 2, 3 species maybe. And this is also corresponding to this trend of progressing from more qualitative methods to highly mechanistic responses of species to climate change. And throughout this whole thing, we are going to be communicating with management, staff scientists, and policymakers, and our overall goal is to make this as useful to the management community as we possibly can. And I have this arrow swooping back to the beginning there from mechanistic to qualitative, and I'll talk a little bit more about that at the end. But our goal is that we can learn a great deal from these more detailed analyses of really data rich species and use that to inform the broader assemblage of species, including ones that maybe don't have great data coverage. So now I'm going to switch over to Verena Wang. Again, she's the postdoc working, doing a lot of the analysis here, and she just started, and she's going to discuss our sample regions and go over this phase 1 qualitative analysis. So Verena, it's all you.
Thanks, Jim. So our project here is going to be focusing on 2 study regions. The first region that we're looking at is the US West Coast, which for this project is the West Coast excluding Alaska. And the West Coast region here is managed by the Pacific Fishery Management Council. And the second region we're looking at is the US Northeast, which extends from Cape Hatteras in the south to the Gulf of Maine in the north, and includes both the New England and Mid-Atlantic Fishery Management Council regions, as well as joint state management that's overseen by the Atlantic States Marine Fisheries Commission. Next slide, please. So the first phase of this project is going to focus on evaluating a suite of species by conducting a qualitative assessment of species vulnerability to habitat constraints. And so this assessment will help us determine if certain life stages represent a bottleneck in species' ability to adapt to climate change. So for this first phase, we plan to assess at least 15 species per study region, focusing on species that are not meeting management projections, and also selecting species that represent a variety of life history strategies. So the quantitative phases that follow this qualitative phase will, like Jim mentioned, will narrow the number of species included in the analyses, depending on the species of interest that we identify here in this first phase, and also based on data availability. So we will be following the framework developed by Pettigat et al. In 2013 in order to conduct life stage specific evaluations of habitat needs and identify potential impacts from climate change. So again, this is a qualitative scoring, and to do this, we'll be using both literature sources and incorporating information from the previously conducted Noaa species vulnerability assessments in order to conduct our evaluations. Next. To walk you through the vulnerability assessment, the first thing that we'll be looking at is the habitat requirement for each life stage. And so this figure here represents an adaptation of the Hardin-Jones migration triangle with discrete physiological habitat requirements for each life stage, from spawning adults and their eggs, to larvae, to juveniles, to feeding adults, and the movements in between these habitats. And these habitat requirements that we're looking at can range from anything from specific temperatures needed for spawning to critical habitat features like sediment type. And so for each of these life stages, a habitat requirement will be scored as either narrow, medium, or broad. Next. The next thing we'll evaluate is the habitat availability for each life stage. And so what we're asking here is basically, how accessible are those previously scored habitat requirements? Are they habitat requirements that are readily available, or are they things that are highly dependent on things like geographic location or seasonal timing? And so the habitat availability that's occurring under current conditions will be scored as either okay, small, or variable, indicating that we don't expect any problems with habitat availability, or that there may be a small and restrictive habitat window that needs to be hit, or that there's high interannual variability in habitat availability. And here in this framework, there's also space to add evaluation for the potential climate impacts on habitat availability. So with the changing climate, will habitat availability increase or decrease, or will it shift locations, or are the outcomes still unknown? Next, please. And finally, we'll be assessing connectivity from one life stage to the next. So this is connectivity, whether by drift of larvae or active migration of adults from feeding areas to spawning areas. And so what we're looking at is determining how important connectivity is from one life stage to the next. Is connectivity something that is not really a big deal and we don't really need to worry about it? It's pretty much assured to happen. Or is it a crucial step where precise connectivity is really needed in order to close the life cycle? And again, there's room in the framework here to assess climate change impacts. And so we'll look at whether the effects of climate change will impact connectivity from one life stage to the next. Next, please. So to give you just a quick overview of what one of these qualitative vulnerability assessments will look like, I'll run through a quick example here using bluefish, which is managed by the Mid-Atlantic Fisheries Management Council. And bluefish are currently below population target levels. So if you look on the left panel here, we've identified 3 potential habitat life cycle constraints that are highlighted in red letters, and 3 potential negative impacts of climate change highlighted in red letters again on the right. So just for example, in the left panel under the 3rd column for connectivity, we've identified that connectivity from the larval phase to the juvenile phase is crucial because larval transport from the outer shelf into estuaries is very strongly influenced by variability in circulation patterns and really influential on recruitment. And in the panel on the right, also under connectivity, we put a question mark there because we've identified a potential vulnerability to climate change because we're really unclear how climate change will impact these oceanographic features and influence recruitment. So these are constraints that we can potentially investigate further in the following project phases. Next slide. So again, phase 1 of the project will focus on evaluating life history stage specific vulnerability of a suite of species in each of these 2 study regions. And I'll hand it over to Lorenzo now to tell you about the next phase.
All right, thank you very much, Verena, and thank you all for coming to this webinar today. So in this next phase, then, those would be the species for which we have identified during the first phase, and for which we have actually access to a rather data-rich scenario, because these next phases are based on spatial temporal analysis of their distribution through different life history stages, and then eventually projection and also modeling of dispersal. So we do need a data-rich scenario here. Next, please. So the part of this analysis will be devoted to understanding whether there are these life history spatial constraints. And so by that, we mean a small portion of the species distribution range that is consistently used over time by specific life history stages, like, for example, the larvae, the eggs, the juveniles, and so on. And here there are 3 iconic examples of species that do have life history spatial constraints, such as the monarch butterfly or the sea turtles or the different species of Pacific salmon. And so then the question is, do we observe similar type of things in the systems that we are targeting for our analysis in the West and East Coast? And if they do occur, these life history spatial constraints, then how can we quantify them, and at what point of the species life history they occur? Next, please. And so as you can imagine, this is a data-rich scenario here because we need different type of data over different life history stages of the species, so different survey type, potentially even different geographic regions, because the area where the species spawn may be different from when they settle and where they move as adults. And so here is an example of where that data sources may come from for the West Coast. What you see on the left, two panels there, that is the consistently sampled grade of the Calcophie region in the South California Bight, which has been sampled since the mid-1950s. And so this would be sources for data such as eggs and larvae. And then on the right side are the footprint of the Noaa groundfish survey in the West Coast in these two different renditions, the triennial survey on the left and then the annual survey that started toward the beginning of the 2000s. To the right. And so through doing analysis over different stages and different size groups of the same survey, then we can look at specific distribution constraints over different life history stages. And this is shown in the next slide, how we intend to do that. Next, please. And so here is an application from the type of analysis that we intend to apply to this data set, but this is an application in the Bering Sea. So this is outside of the scope of this study and also the geographic region that we propose to apply this analysis for this study. But it's illustrative of the methods and the type of quantification that we can get from this analysis. So in this particular case, this is the Eastern Bering Sea shelf. This is Alaska Place, which is a small flatfish, which is mostly shelf-oriented. And these are, what you see there are the different distribution patterns going from different, progressively older and larger life history stages. So on the upper left panel, it's the distribution of the eggs, and then immediately after is the distribution of the larvae over multiple years, of course. So the average distribution. And these are from Ichthyoplankton survey. And then progressing more toward the right, and then eventually the second row of plots, then there is the distribution of progressively larger life history stages, and you can see the size range on every plot. In addition, in each of these plots, you also see the R-square, which is the percentage of spatiotemporal variance explained by this relatively simple model, which is at the bottom of these charts there. And that is a relatively simple spatiotemporal model because it only includes the spatial coordinates, so that latitude and longitude. And so the rationale here is that if this very simple spatiotemporal model can capture a large portion of the spatiotemporal variance of the species at that stage, then it means that that area has been consistently occupied over time. It's always going back to that place over and over again. So just having spatial coordinates is good enough to represent their process of distribution. And so if we look now at the pattern of these charts, then you can see that for this species, again, they start off relatively highly constrained with more than 50% of the variance explained. And then as they grow, they tend to expand their habitat. So they grow, they gain greater morbidity, they move farther away. And also the variability from year to year increases because this simple model actually is losing its explanatory power. And this is indicative of other processes that may affect the distribution which are not included in the analysis. And of course, the most obvious one would be temperature. And so it's indicative. So this R-square is also indicative to the degree of consistency to which that habitat has been used over time. So we intend to apply similar analysis to the species that we're going to target both in the East and West Coast. Next, please. So this is again an illustrative example of what we see when we pull together results from multiple different species. So results of the analysis are just shown to multiple different species. This is a meta-analysis of 10 different species. On the y-axis, there is that magic of habitat constraints, so that percentage of the spatiotemporal variance explained by that very simple model. And on the x-axis, there is an ontogenetic index going from 0 to 1. So it's a relative index simply expressed as the target size or age divided the maximum size or age. And it's a way to put different species with different life span on the same axis. And so we can compare this pattern, average pattern across multiple species. And the picture that emerged when we have done that for subarctic species, so species that are not yet in the system in which we are about to start, is that there is this average pattern, significant pattern, where the species start off relatively constrained in space and time. So those would be the spawning aggregation, the larval aggregation, and potentially into the juvenile habitat. But then eventually that level of consistency of spatiotemporal constraint decreases over the life stages of the species, and that because probably the species gain greater mobility and it's following other environmental gradient. And so the other part that is also interesting in this plot, that even though we do have a significant relationship, that relationship has a lot of variability. It's really depending on the species. So different species may be doing different things. And while that is adding variability to this plot and therefore challenges, especially from a statistical point of view, from an ecological scientific point of view, that is really the interesting part, because it allows us to make those connections between what each species is doing and the life history traits or strategy of the species in order to make this expansion from data-rich cases to data-poor cases. And so this is shown in the next slide, please. And one more, because there is an animation. So if you could hit, yeah, perfect, thanks. So these are the 10 case studies that are included in that scatter plot I just showed before. And these three different patterns are emerging from looking at them individually. On the upper left, in that blue circle, are the species for which there is no significant relationship between the habitat constraints and the ontogenetic index. And those species are at the northern range of their distribution and in the habitat in which we have been looking at them. And so they may be particularly sensitive to changes in temperature and therefore have more of that spatial distribution related to temperature, which, remember, was not included in that model. Then moving on to the next group, which are essentially part of the top row and the first two panels in the bottom row. Those are the species for which there is this significant relationship represented by this overall decrease in that matrix of constraints over the species life history. So those are the species start off relatively constraints, and then eventually they gain mobility and increase spatiotemporal variability over their life size. And all of these species are the gaddites. So there is a commonality in their life history strategy in which, in the way in which they use their habitat. And then the last group, which is the one encircled by the green ellipse there, those are the species for which we see a relatively strong relationship, and also that relationship that remains toward the later life history stages. So moving into the juveniles and potentially even in the sub-adult, as it is shown in the latest panel. So these are species for which there is a strong affinity to their habitat characteristics. And all three of these species are small flatfish, shelf-oriented flatfish. And those are the species for which it would, at least conceptually, would be reasonable to expect that there is a strong link with their habitat, because their diet is linked to the sediment characteristics and to the epibenthic and infaunal organisms which are in these sediments. So that type of consideration are the one that we're trying to exploit and leverage on in order to make this type of connection with life history traits. Next one, please. So what does that all mean at from the point of view of the species adaptability to climate change, and also for the rest of the analysis that we plan to do in this project? And again, this is shown by way of example. This is a study from Stevenson and Loud that came out in 2018 that shows the distribution of Pacific cod in the Bering Sea in relation to contrasting temperature years. So on the upper panel is the footprint of the bottom temperature in 2010, which was a relatively cold year, and then 2017 to the right, which is a warm year, representative actually of a number of different years in the latest time frame. So 2017, 16, 18, 19, they've all been warm. And on the bottom panel, then there is the distribution of cod from the survey in these 2 years, and expectedly what you see there is that during these cold years the distribution is more outer shelf and southerly oriented, whereas in this warm year there is this radical movement toward the north, so that the majority of the biomass actually, at least during the time of the survey, is in that upper portion of the inner shelf. So but in the meanwhile, their spawning habitat are linked to topographic feature, and for cod we know that their spawning aggregation are at the Bering Sea slope edge. And so those are those shaded region at the edge of the shelf. And so that poses the question, if there is this continuous warming and if there is this tendency for the adult portion of the population to move far to the north, will they be able to then close the loop and come back to where they need to be in the winter in order to spawn? Or will they eventually spawn where they are because the distance at that point is greater? And if they do spawn at that point in this newly occupied habitat, will the species then be able to successfully drift the eggs and the larvae into their juvenile aggregations? And so this is the case. These are the exemplative of the case studies where we intend to go then with this projection in order to identify potentially new habitat that a portion of the population is going to occupy, and then eventually with the larval connectivity model in order to see whether hypothetical new spawning activity from this new habitat would lead to successful recruitment into the nursery areas where they are known to reside during the juvenile phase. And with that, I think I conclude this session, and I will, this will lead on to Mark for the management context. Thank you.
Thanks, Lorenzo. Yeah, my name is Mark Nelson, and I'm going to try to now to provide some context on how our project fits into fisheries management and how it can be used by the decision makers to better meet management objectives. So for the most part, fisheries are still being managed with static state models. These, these models use historical data to develop forecasts for future abundance. However, we are seeing more and more of our fisheries shifting and changing, and we realize that the ecosystems that these fishery resources reside in are not static. So we have empirical evidence for many species that indicate long-term directional changes are occurring in their life history and their population dynamics parameters. Of course, these changes impact the reference points that we use for stock assessment in setting sustainable harvest targets. OFL, ABC, ACLs. But at the same time, these stocks are shifting in space due to them trying to maintain their climate envelope. And these shifting populations can confound the population abundance trends, especially when the stock moves across, sampling ranges or stock assessment boundaries or even management boundaries, including international boundaries. So understanding if abundance trends are due to population dynamics or due to the species shifting and moving out of an area is really critical. Next slide, Jim. So Noaa has been trying to address these issues in a number of ways, including improving and increasing federal surveys and developing best practices in species distribution modeling, such as DISMAP. But some scientists are also using historic range shifts trends to develop forecasts for future shifts, but this really has only been done for a small group of species. In addition to all this, Noaa has also concluded qualitative climate vulnerability assessments for about 450 fish and shellfish stocks across all US regions except for the US Caribbean. And in addition to that, Noaa is completing a qualitative climate vulnerability assessment for 52 marine, estuarine, and riverine habitats located in the Northeast US. And whereas these findings from these qualitative assessments are highly accessible to policymakers and stakeholders, the results give a relatively coarse-grained understanding of the mechanisms driving the vulnerability. So the proposed work that we're doing is trying to build upon these studies and then fill in the gaps left by these studies, and trying to move the the state of the science forward. So next slide. By developing a model to determine a species' ability to track climate velocities across all their life stages can help provide managers a more insightful mechanism for characterizing the species distributional shifts. And this should be able to help managers anticipate future issues with these shifting species. By giving the managers and decision makers better tools to understand the potential population shifts, gives them the context that they need to go through the difficult process of reassessing the current allocation of catch between fishing sectors and or states. So this could allow managers to develop proactive management decisions that don't lock management objectives into a historical state that no longer exists. And another thing that I want to make sure that we don't overlook, that these assessments and understanding shifting species shifts can also be used by the fishing industry, including fishermen, processors, and other related businesses. They can use this information to help inform their business decisions. So this could, for instance, this could influence a fisherman's decision to buy, sell, or transfer quota, maybe buy a bigger boat, or maybe to get out of a particular fishery and enter a different one. Land-based processors could also use this information to help inform business decisions, such as making long-term plans for infrastructure investment or employment employment decisions. So overall, there are a lot of potential audiences that this project could help inform. So with that, I think I'm going to be passing it over to Jim to wrap up the presentation.
Jim, I think you're muted.
Communicating. Hopefully you can still hear me.
Yeah, we hear you now.
Okay, sorry about that. I heard some beeping and things. Okay. Communicating with management and the scientific community and conducting a qualitative analysis. So we're in the midst of all that presently, and we're planning a trip to a Mid-Atlantic Council meeting and a Marine Fishery Commission meeting later this year if things open up. Year two, we'll be in the midst of this spatial modeling analysis across life stages and starting to format climate projection data and calculating climate change exposure across life stages. Plan a Pacific Council meeting that year, and in the final year, we'll be conducting some projections, life stage specific projections for a handful of species, and using a larval transport particle tracking model to examine present and potential future disruptions of population connectivity or life stage connectivity. And then we'll be finishing up with a decision tree, which I'll talk about in just one second. And then again, another trip to a council meeting and a commission meeting in year three. So just to cap things off, we are going to attempt this decision tree framework. This graphic is, a very crude representation of what it might look like. But the idea here is that we want to explore if we can use really widely available information, and these are things like historic distribution shifts on web portals like Ocean Adapt, species projections, climate vulnerability assessments, and basic life history traits, to see if we can identify vulnerable stocks, prioritize and focus research goals and potential data collection problems, and ultimately improve methods for future distribution forecasting. And again, our hope is that what we learn from the more detailed mechanistic analysis on the really data-rich species can be helpful to inform on the broader assemblage of species, including species that we don't have a lot of as much information on through this decision tree framework. Again, here's the project team, and that's it. And happy to listen to questions. And thanks again for listening.
Awesome. Thanks, everyone. Thanks to all of our speakers for your great presentation. I know that this is a complicated topic, but I really appreciated the way you broke down your research approach and made it pretty easy to follow. So I'm going to go ahead and take back presenter privileges, Jim.
Okay.
And just share my screen, which also has contact information for the speakers today. So if anyone is interested to follow up with them, their information is right here. So we have a couple of questions that came in during the presentation. So I'm gonna just go ahead and dive right into some of those questions. And I will just mention to attendees, there's a Q&A panel and there's also a chat box. And I'm mostly following the Q&A panel. So if you put a question in the chat box, I'm trying to monitor that as well. But I'm most likely to see it if you do put it into that Q&A panel. So just a quick tip for our attendees that are trying to ask questions. So our first question asks, how will you ground truth the outcomes of the initial qualitative portion of the project? And are predator-prey interactions and dynamics included in your definition of habitat to address and incorporate ecosystem-level dynamics in the evaluation?
This is Jim. I'll take a stab at that one. I'll answer the second part first. Predator-prey interactions were very much in our discussions as we were developing this project. But yeah, I think ultimately we decided that we had to kind of cap the analysis somewhere. So we don't have plans to explicitly include any predator-prey dynamics, although they might be some of the reasons why species have the habitat associations that they do. But yeah, we definitely recognize that predator-prey dynamics are a key aspect of species climate response and maybe an even more difficult one to address. And then the first part, I'm trying to see if I can read what you said, ground truthing the qualitative analysis. I think in a sense that's kind of the point of the decision tree, and to see if we can do these qualitative analyses with the broadly available information without conducting any major analyses on this 30 some species that we'll do the qualitative analysis on. And if, as we progress through the more mechanistic aspects of things, if we see patterns in the species life histories that relate back to those qualitative analyses. I mean, it's possible that we won't see anything, but based on work that Lorenzo presented with the species spatial associations and restrictions and how they were able to relate those to some of the life history traits, we think there's a strong chance that we will see we will be able to relate back to just the species with less information on, and that it will be useful, but we'll see. Hopefully that answered your question.
Great, thanks. And our next question asks, in data-limited fisheries, information or data on a given species are often pulled from studies in other geographical regions, or even studies of different but similar species. I'm wondering how broadly applicable or transferable you foresee your findings being.
Maybe I'll take an initial stab at that. This is Lorenzo, and then anybody else could chime in. But yeah, that's a great question. Sometimes we see even same species over different environments doing completely different things, even though they may be closely related taxonomic and evolutionary to each other, or maybe even in their ecology. I think we need to have we just need to apply, careful consideration when we get to that point. Like one example that I have shown in those plots before is the case studies where the species at the northern end of their distribution were not really exhibiting any particular spatial constraint, at least through the life stages that we have been examining. And that is indicative of, a species that may be sensitive because of the geographic location, not necessarily because of life history strategies or traits. So they're at the northern end, they may be particularly vulnerable to changes in temperature and therefore more responsive to that, as similar populations which are perhaps at the core of their distribution. So we will need to keep in mind that and make those extensions with this consideration in mind. A great question.
Awesome, thank you. And our next question is asking about adaptation policies. So, I am wondering if anyone on any of our speakers might be able to answer this, but they're asking about what are the key requirements to develop sound adaptation policies across scales? So for example, 1 to 2 key challenges and 1 to 2 opportunities to translate this science into applied adaptation strategies.
Maybe I'll take a stab. That's a great question. In the way of challenges, I can think of many. I'm thinking of this, at least at this moment, more from the computational side. And so the data availability is definitely a big one, especially for this analysis, data-intensive analysis, where we try to look at different spatial-temporal pattern of different life history stages. Even though we may have data for different life history stages, there are all sorts of issues that come to play. Are we actually sampled the entire range of distribution? Are we actually sampled on the right time of the year? And so on, into this rabbit hole of data analysis. So that's on the side of challenges. On the side of opportunities, I think there are opportunities for adaptation strategies related to the way in which we monitor certain species and habitat that would, for example, highlight data needs that could be filled with either existing surveys, maybe slight modification of existing surveys, like modification in time, the time in which we go out, or the extension of the survey. I'm thinking about that code examples that I showed for the Bering Sea. One outcome of those recent analyses that show this northward projection is the need to go and sample farther to the north to look at what that population is doing in that part, because that has implication also for the assessment and the catchability of the species within the survey area. And so that to me seems to be an adaptation strategy, at least in the way we monitor and assess the species that has come out of this type of analysis.
Awesome, thanks. And if anyone else has things to add, go for it. But I'm gonna go to the next question. Are you, and if so, how, are you utilizing the scenario planning work being conducted by the Pacific Fishery Management Council in the modeling you are doing?
This is Jim. Lorenzo, are you familiar with the scenario planning?
I'm not. Well, but that doesn't mean that we will not follow that path. So we're that would be definitely an area where we need to look into and work in coordination with what is being done for this scenario. We do intend to connect with colleagues from the West Coast Noaa group, and therefore these opportunities hopefully will emerge as we move on. But if there are more direct links that you foresee, please don't hesitate to get in touch with us.
Excellent. Yeah. I'd encourage the person who asked that question to please reach out to us after the webinar, just so we have that in the records and can keep conversations going on with that. So the next question asks, will not having larval surveys in other regions compromise conducting similar analyses in those regions?
I can cover that, I think. The larval survey, as I understand it, is pretty comprehensive in the Northeast, the EcoMon survey, which I think will be useful. So perhaps the question is directed more to the West Coast, where I think Cal Coffee might not extend all the way up the West Coast. I think we just have to be careful in the species and the examples we use. I think that to analyze each individual species, we don't necessarily need to have every single life stage covered. So some species we might have excellent data on juvenile and adult stages, and that can help get at some of these questions while taking advantage of the species that do have good coverage for larval data.
Great, thanks. All right, we just have all these questions coming in. This is great. I'm really excited to see a lot of engagement from this presentation. So thanks, everyone. So our next question asks about how will you quantify the overall fisheries benefits in terms of recruitment and survival based on the implementation of adaptation policy across various temporal and spatial scales?
Well, I don't know that we're running any like a management strategy evaluation or trying to assess how adaptive policy might affect or how might different species distributions be affected by or effective or less effective from different adaptation policies. In terms of estimating recruitment and survival, I think some of the trends we might be looking at are shifts in where recruitment is successful and perhaps understanding or making some better links between recruitment patterns or shifts in or expansions of abundance of early life stages and relating that to potentially biomass and production from farther offshore. I think we'd be able to address some of those questions with these data. However, we're not necessarily running simulations of population dynamics simulations through these analysis and estimating changes in production per se.
Excellent, thank you. Well, it looks like we're getting really close to the end, but I will take, there is one more question that hopefully we can wrap up right before we have to get off. So this is a question for Lorenzo. And this person says, thanks for the contribution to this interesting presentation. Could you recall the difficulties that the team had to compile the minimum data requirements to conduct the research on the range shifts? And I believe they're referring to the data that you presented, Lorenzo, for a related project. And then adding on to that question, they're curious if you can provide advice to this in regard to countries with poor data sampling in fisheries and independent scientific surveys, especially where those fisheries are economically important sources at the national and fishing community level.
Great, thanks for that question. The requirements is that of having, from the data side, multiple years of data for different life history stages. So ideally also the egg and larval portion, because if there is an egg phase, because that is revealing of the spawning aggregation. But the spawning aggregation can also come from the mature individual during the spawning season. And for multiple years, I would say at least 4 or 5 years of continuous this type of survey. But on the extension to other case studies, I think fishery dependent data can be very useful. Expert knowledge could also be very useful to do the similar type of analysis, at least in a presence absence type of framework. And so that would be an area where I would look into. Thanks.
Great, thank you. All right, and with that, we are right at 3 p.m. East Coast time. So I just want to thank all of our speakers so much for your presentation and for sharing this research with us. Really looking forward to following along and seeing how it progresses throughout the lifespan of the project. And thanks to all of our attendees for listening in and also for your great questions and engagement today. We really appreciate it. And with that, we'll go ahead and wrap up. And thanks again, everyone. Also, don't forget about the really quick survey that will happen when you close out of this webinar. If you could just take a few minutes to answer some of those questions, that would be really helpful. And also feel free to follow up with any of the research team or anyone at LenFest if you have further questions or conversations that you'd like to have. And with that, thanks again. Have a great day, everyone. And hopefully we'll be hearing from you. Thanks. Bye.